अब हम बात करेंगे प्रोसीजर की फॉर हाइब्रिडाइजेशन। हमने रिएजेंट, सप्लाईज, हाइब्रिडाइजेशन सॉल्यूशंस के हवाले से तफसीली बातचीत की। नाउ वी आर गोइंग टू स्टार्ट द हाइब्रिडाइजेशन प्रोसेस, हाउ टू प्रोसीड स्टेप बाय स्टेप। जैसा कि पहले मैंने जिक्र किया कि हमारे पास 3 किस्म की ऑप्शंस हैं टू डू द हाइब्रिडाइजेशन, ईदर इन हीट सीलेबल बैग, in roller bottle or in plastic container. We are going to use a roller bottle for the rest of the procedure. This slide shows you the flow sheet of the procedure, the steps which are involved. The first step would be to make the membrane wet using a buffer and then proceed for pre-hybridization for a given period of time using a specific pre-hybridization solution. Then proceed further for hybridization, washing and autoradiography. In the washing step, we will use 3 or 4 different kinds of washing solutions, and then we proceed further for autoradiography. To make the membrane wet, we will use the buffer or the solution 6S SSC, that is the sodium citrate and sodium chloride solution, or in place you can use the phosphate solution as well, 6 times SSPE. And that will be visible that how much time is required to make the membrane completely wet. Somewhere around it takes about 2 minutes. Once the membrane is wet, we have to proceed for pre-hybridization. As we are going to use roller bottle, so हमें अपनी membrane को fold करना है या roll करना है to make a cylinder like structure which can be pushed into this roller bottle. For that purpose, we have to roll it up very carefully that we don't break the membrane, otherwise we are going to lose our whole of the work. उसके बाद इसमें हमें pre-hybridization buffer की addition करनी है, and that is going to be again the same 100 microliter per square centimeter of the membrane will be added in this roller bottle. अब ये roller bottle जो है ये different size में available हैं, 6 inches, 12 inches are available. So we have to see that if we need 6 inches bottle or we need 12 inches bottle. For 6 inches, then keeping in view the size of the membrane, we proceed further. एक चीज़ का यहाँ पे ख्याल रखना है, प्री हाइब्रिडाइजेशन में जब मेम्ब्रेन को हम इस रोलर बॉटल में इंसर्ट करेंगे, देयर शुड नॉट बी एनी एयर बबल इन बिटवीन द ग्लास ट्यूब एंड द मेम्ब्रेन, because if there is an air bubble, it is going to interfere with the hybridization process. Once the pre-hybridization solution is added in the membrane is inserted into the roller bottle, then we have to place it at an appropriate temperature for 10 minutes, 20 minutes or 30 minutes. It has to be optimized कि हमें कितने टाइम के लिए इसको incubate करना है. अगर हम aqueous solvent यूज़ कर रहे हैं तो temperature will be 68 degree centigrade. अगर हम formamide containing solvent या solution यूज़ कर रहे हैं then it is going to be 42 degree centigrade, and for phosphate SDS solvent the temperature will be 65 degree centigrade. Again, ये temperature जो हैं they are not fixed temperature. You can optimize the temperature by increasing it or by decreasing it wherever the best result is visible. During this time period, when we placed the membrane for pre-hybridization, we proceed further to denature the DNA. And for that purpose, the probe, which is radio-labeled double-stranded DNA, we have to denature it so that the double-stranded DNA, it gets separate and the single strand will easily bind with the DNA immobilized on the membrane. For that purpose, we have to heat the DNA at 100 degrees centigrade for 5 minutes and immediately chill it in ice cold water so that renaturation will not be possible. The next step would be hybridization. All of the process is going to be followed which we did for the pre-hybridization. The only difference will be now the buffer contains the probe. For pre-hybridization there was no probe. For hybridization there we add the probe. For this purpose we are going to remove the pre-hybridization solution and we are going to add the new hybridization solution and placing the bottle back into the hybridization oven for the specific period of time with the constant rotation. Once the hybridization time is over, now this is the time to wash the membrane so that any unwanted, any unincorporated probe should be washed off. For that purpose, there are two possibilities: either take out the membrane, place it in a tray, or wash the membrane in the roller bottle. For that purpose, remove all the hybridization fluid, discard it in a proper radioactive waste container, and then use the washing solution, which is SSC and SDS. We have three different solutions. We have to see that which solution is going to be added first and then later on. Several milliliter are going to be used. Here we can recommend 1 milliliter per square centimeter of the membrane, and constant rotation or agitation is recommended. From washing onward, membrane should never get dry. Otherwise, the problem will be there for the results. After 5 minutes of this agitation washing, replace the buffer with the second type of washing buffer, SSC and 0.1% SDS. Again, 15 minutes incubation with agitation. Then discard the washing waste and then add the third washing solution, that is 0.1 x SSC and 0.1% SDS. Now keep it for 30 minutes for 4 hours, from 30 minutes to 4 hours at 65 degrees centigrade. And at the end, simple rinsing with the 0.1 x SSC. Once the washing is done, take the membrane out, place it on some paper towels so that the excess of the liquid is drained off or dried, and then pack it in the cling film or the very fine plastic sheet. Wrap it in that cling film, and then we can proceed further for autoradiography using a specific cassette. Place the membrane which is packed in the cling film, place the radioactive film, close the lid, and then place it on the minus 70 degrees centigrade for overnight or for the required time interval as per the sample or the process we have discussed earlier.